As you guys have been noticing, one of the themes of this chapter is comparing the process of mitosis that we talked about last week with the process of meiosis that we're discussing this week. In the process of meiosis, so far we've done meiosis 1. So we did prophase 1 where synapsis occurred and crossing over happened to give us that source of genetic variability. In metaphase 1, we had independent assortment, which is our second source of genetic variability. In anaphase 1, we separated the homologous chromosomes to become haploid. In telophase 1, we did cytokinesis and reformed those nuclear envelopes, and now we have two haploid daughter cells that are genetically distinct from each other. Now, during mitosis, we would enter the cell cycle again, and we would go through the process of the G1 phase, the S phase, and the G2 phase. In meiosis, the point of this process is to take that diploid cell and turn it into haploid cells. If we do this DNA synthesis, this S phase right now, during the interphase of meiosis 1 and meiosis 2, that would defeat the whole purpose of having our genetic material. So we're going to shut down S phase. So no DNA duplication during interphase between meiosis 1 and meiosis 2. All right, so now we've did all meiosis 1. Let's get into meiosis 2. Meiosis 2 looks like mitosis, except for we have half the DNA. Okay, we're still going to have the process of prophase, metaphase, anaphase, and telophase. And of course, we're going to use the last name of 2 so that we can keep all that straight. Okay, so in prophase 2, we're going to undo what we just did. We're going to break down the nuclear envelope. We're going to form new spindle fibers, right, so that we can attach those sister chromatids and pull them apart. We're going to take our DNA from loosey-goosey spaghetti form and pack it up into Cheeto form, right? We're going to form those chromosomes again. Okay. In metaphase 2, we're going to line up our chromosomes along the equatorial plane, just like we saw in mitosis. Our spindle fibers are going to attach to each of the sister chromatids. So then in anaphase 2, our sister chromatids are going to separate just like we saw in mitosis. In phase 2, we're going to reform that nuclear envelope around the haploid nuclei. Now we have done all of meiosis. We've done prophase 1, metaphase 1, anaphase 1, telophase 1. We did prophase 2, metaphase 2, anaphase 2, telophase 2. So what do we have at the end of this? At the end of meiosis, we have 4 daughter cells. They are haploid, and they are genetically distinct from each other and from the parent cell that we started with.